Hi everyone, my name is Arita Lopez. I am very excited for today's demo that I'll be doing with you. So today, just as everyone's logging in, we're gonna be working on a lava lamp today. So with this experiment, we will be exploring density and polarity. And as we are presenting or going through the presentation, if you would like to participate, that would be super awesome. And I do appreciate it. So just start jogging your memory if you know anything about density and polarity, if you'd like. Okay, so as you saw in the email, you did get the material list. So we can just go right through this now. So we'll be conducting the experiment right now, and then afterwards we can just talk about what we saw. So I'll go ahead and just stop sharing now so you can see what I'm doing. Just a second. All right, so what you want to have is basically a nice working area that you can have at home. What I did was I just laid out some Kleenex here, and I made sure that, I'm not going to stain too much. So what I'm gonna do is I'll actually grab my water bottle here. And what I'm gonna do is I will grab the vegetable oil that we have here and fill it up 3/4 full of the bottle. So I think of it in pieces of 4 equal parts. So I just fill up 3 of those 4 parts. And as we are going through this, just you can comment or ask yourself any questions that you might have, or just anything you'd like to share as you're seeing what's happening. Okay, that should be about right. And after that, we will be adding water to the neck of the bottle. So you can leave a little bit of space between the water line and the top of the container. That's fine. So already we're seeing a lot of change with the mixture here. So feel free to just drop anything you see, or make note too if you'd like, just to see what the difference would be later on. So after that comes the more fun part of it. So you can also just at this point decide on a color that you'd like. I'm actually using green. If anyone has questions or is having trouble, please let us know. So I'll be adding about 6 to 8 drops or so. It's probably my favorite part of it. And then after that, they actually just set down, and then after that you will be grabbing your Alka-Seltzer tablets. So I actually just cut mine up into 6 pieces, and as I drop them, I'm going to just let them, do their thing. And there will be a reaction here. So I just want to do it kind of slowly as well. And just a fair warning, at this point, the water might start bubbling up. So as we're adding these, before sealing everything, we want to make sure that the bubbles stop so that we don't, make it explode with the cap on. And this happens fairly quickly too, and you can also see the colors changing. You can feel free to ask a question. I'll try to look at these questions right now. Oh, and it's actually bubbling up right now, so I can show you guys. Just waiting for it to kind of calm down. So this is why this is very helpful to have extra towels next to you. You don't want to stain yourself with oil. That wouldn't be fun. So you can see a little bit more of it. I have one more to drop, so just wait for that. And maybe I'll clean it up too. So it could get oily.
Great. So we have a question in the chat. What would happen if you put on a cap?
The pressure will build up, and basically it'll just, get a little bit crazier. So right now it's even just spilling over, and that happened to me actually. I did it not that long ago with everyone or a couple people here, and it was very hard to contain. So it was just spilling through the cap as well. That was a nice question, by the way. Does the ratio of water to oil affect the reaction after you put in the tablets? I'm not sure. I would think it would, do the same amount of the reaction. I just think it needs enough of both to be able to do that, but I think it would probably more of like a smaller scale. I don't know if anyone else knows exactly if it affects it differently. These are really good questions. Where do the bubbles come from? So just to put it in short, there is part of it is carbon dioxide is created. So we'll touch a little bit more on that. But really good questions. So after this, you can actually grab your bottle and you can actually just shake it. You can choose if you want to like twirl it or shake it or go up and down. But at this point, I can actually just explain what happens with the Alka-Seltzer and why there's bubbles. So it's because the Alka-Seltzer is reacting with the water. So that's what makes the carbon dioxide gas bubbles that you were talking about. And they stick to the water droplets. So this is like a water and gas combo. And it's the water and gas combo is less dense than the oil. So then that's when they rise to the top. So you can see how they're trying to set. And then I'll go ahead and share actually after this, now that you've seen this, I'll actually add in where we talk about the polarity and the density. So let me share my screen again. And there's a small little 1-minute video.
Notice that oil floats on top of water? It has to do with how water molecules interact with the oil molecules. Oil is insoluble in water. That means it doesn't dissolve in water. The way in which two substances interact depends on the molecules that make up the substances. When oil is poured into a glass of water, the water molecules and the oil molecules meet for the first time. As they are stirred together, the water molecules and oil molecules move around each other. When the stirring stops, the oil and water immediately begin to separate. This is because the oil molecules are not attracted to water molecules. They are more attracted to other oil molecules, and so the oil molecules clump together. At the same time, the water molecules also clump together. Water molecules are more attracted to each other than they are to the oil molecules. As the oil molecules and water molecules clump together, they separate. This is because oil is less dense than water. The oil molecules rise above and float on the water molecules. That is why oil and water.
So I'm just gonna change to the next one. since we already talked about that. So if you, again, have questions as we go, feel free to share. And then this is just with explaining polarity. So if anyone has any questions about density or the video we just watched, we can go through that right now. And then I'll go ahead and just explain this file. So as you can see, there's two bears, and one of them is like, help, help, I'm dissolving. And then the other one's like, but bears are insoluble. And then the polar bear is saying, that's easy for you to say, you're not polar. So if you have any idea what polarity has to do with, basically what it is, it prevents the oil and the water from mixing together. So kind of like thinking of a magnet, it's kind of like opposites attract, like north poles are attracted to south poles. So the positive end of a water molecule will connect with the negative end of other molecules. So oil molecules are non-polar, so they don't have a positive or a negative charge. So this is why they don't mix with water. Oil and water don't mix. So at this point we are done, but let me see if we have any other questions here. Yeah, I really like this little meme as well, or comic. How does the emulsifier mix the oil and the water? I would think it's from the movement that you're making by shaking it, and so it also has to do with the density of the oil and the water. And that is all for today. If anyone wants to, like, honest, if you guys have the lists, I do encourage you if you want to do it later on and show like your friends and family. But I hope you guys enjoyed it, and thank you everyone. I hope you guys enjoyed the rest of the talks today.
All right, thank you so much. Really excited to introduce to you this week's speaker, Arita, and she is a graduate student in genetics, and she's going to be talking to us about how she studies birth defects and the genes that cause them. So I'm now going to hand the floor over to her. Please give her your full attention. Yeah.
Hi, everyone. So I'm Arita. I'm studying for a PhD here at Yale in the genetics department. And so today I'm going to tell you a bit about what my research is on and what it means for real life people, and what kind of animals and different experiments I do. So before I get into the science side of things, I just want to tell you a little bit about myself. Okay, so about me, I grew up in Phoenix, Arizona. So as you can see, here I am eating a snow cone to cool down from the desert heat. I then also spent part of my childhood in India. And then after that, I went to university in the United Kingdom. So this is me at my graduation ceremony. We had to wear very ridiculous outfits for the ceremony, and it was very hot in this fur hood. But it was fun anyway. And when I was in the UK, this was when I first became interested in the type of science that I do, called developmental biology. And back then, I used to work with an insect. So this is a species of fruit fly. So in your kitchens, you might see if you have any fruit lying around, there are these little flies that are attracted to it. So I used to actually work on these flies and try and find out how they make their eggs. And so after I did a lot of work on that, I came here to New Haven, to Yale, where I've been working on something a little bit different. So let me tell you about that now. So I work on how a body can tell which side is left and which side is right, how that decision is made. So let's go on a little bit to talk about why this is relevant and why this is an important topic to study. So my lab is actually interested in studying birth defects. So about 3% of babies born every year are born with some kind of birth defect. And actually here in the United States, birth defects are the leading cause of infant death. And this is obviously extremely tragic when parents have a child, they hope that this child is going to be happy and healthy. And then if they find that this baby is born with a birth defect, this can cause, the baby a lot of suffering and pain, but it also causes the parents a lot of suffering and pain as well. And one thing that makes this situation very difficult for everybody involved is that a lot of the causes of why babies are born with birth defects are actually not really known. So a lot of birth defects are caused by environmental causes. So, maybe the mother had some kind of chemical that caused a birth defect or something else. But actually, most birth defects end up being caused by something in the baby's DNA, something in their genetics. And most of the time, we don't know what is in this baby's DNA or what is in their genetics that's causing this birth defect. And this can be difficult for doctors because if they don't know why this birth defect happened, it makes it more difficult for them to treat it. And it makes it difficult for parents because often parents want to know if they have any other children, might they be affected? So in my lab, we like to think about this problem. And we ask the question, how can we find the genes that are needed to create a healthy, happy baby? So perhaps we can talk about this in the chat. So do any of you have any ideas of how one might find things that are needed, like what is needed for a baby to be happy and healthy and not be born with a birth defect? Anyone have any ideas of how we could try and find that stuff out? So I think one person said interviews. So yes, so that's a really important part of figuring out what's gone wrong with when a family has a child who's born sick. We will talk to their parents, we will talk to the parents of the child and find out what's been going on in their environment. That is a big contribution to this. Family trees, yeah, for sure. So that's also very important. We can look back at other members in the family and find out whether they also had birth defects or not. And this can tell us whether this is something genetic that runs in the family. All right, yeah, so those are some good, decode the human genome and find deletion. Yeah, I think that's our ultimate goal, right? We want to know what every part of the human genome is doing and see which parts of it can go wrong to cause these kinds of problems. All right. Okay, so I'll tell you, there's many different ways that we can do this, but I'll tell you a bit about how my lab goes about trying to find the genes needed to create a baby. So the way my lab does it is we use a method called patient-driven gene discovery. So in this method, we try to find patients out in the world who have some kind of birth defect that we're interested in studying. And we'll approach these patients, we will collect tissue samples from them. So we'll collect cells from their cheeks, have them do a cheek swab, we'll take blood samples, and from these samples of their tissue, we can take their DNA and sequence their DNA to try and look at their genes. And then we have scientists in our lab who analyze this data that we get from this DNA, and we then turn that DNA sequence into a number of genes, and we can see which genes have mutations in them. And so once we have a list of mutations that are in the baby, our job isn't over. Every human on Earth has many mutations in them. Not all of them cause health problems. So we need to actually find out which mutations on this big list of mutations that we get from baby are actually causing their health issue. So the way that we do that is we actually use animals in our lab to test these mutations. So there are many labs here at Yale that test mutations that we find in patients, in animals such as mice, in bugs. We create these mutations in those animals and see if they have similar kinds of health issues. And once we found that a mutation can cause a birth defect in an animal, we can then use this animal to start asking further questions of what actually is this gene, or what actually is this mutation doing that's resulting in a birth defect. So this kind of approach, lots of scientists around the world who study this kind of thing have their own approaches to do it. And all of these approaches have their own pros and cons. So this approach has its own pros and cons. So the pros are that since we found these genes in patients, in real life human patients, we can be sure that these genes actually do play a role in the human body. Sometimes a lot of other methods, they find genes in animals first. And in that case, we might find genes that are important for the animal, but we don't necessarily know if they're important for the human. So this way we know that they're important for humans. And additionally, even if we're not that interested in human health, even if you're just interested in science generally, finding these genes in both human patients and in animals tells us that the gene you found is really important for a huge group of animals rather than just one. But this method sounds great, but there are some. It's actually very difficult to do. So that's the main con. It's quite difficult, and the part of it that's the most difficult is this first step of trying to recruit patients to your study. So in order to find a patient that you can recruit to your study, you need to know doctors who treat patients and hence can tell you about them. You need to have access to hospitals that the patient can go visit and be treated in. And not only that, a lot of times these patients they've been through a lot. They've had lots of medical struggles. They've been to hospital and doctor after doctor, and sometimes their parents have lost trust in their doctors to find out what is going wrong with their child. So we have to gain the trust of these parents by being very careful with their children. So seeing as this is the main part that's difficult about doing research like this, here at Yale is actually a great place to do research like this, because here at Yale New Haven Hospital, we have a program called the Pediatric Genomics Discovery Program. So this is actually a program that has contacts with doctors all across the world, and doctors who treat patients who have birth defects, like the ones we're interested in, can refer their patients to our program. We then have our own doctors and scientists who come meet the patient, meet their family members, hear all about their story, and then we can collect DNA samples from them. And then those DNA samples come to scientists like me in the lab, and we analyze those DNA samples and try to find answers about what is going on in this child, what gene might be causing their problems. And then once we find something, if we manage to find something using our experiments and stuff, we can then share these discoveries with the family of the patient, and that is often helpful for their care. So I'm going to tell you a bit about the project that I'm working on. And so this is a nice example of this entire process and how we can start off with a patient who has some kind of health problem, and then how their problem can actually help us find some interesting science. So I work on a gene called synaptotagmin 8. It's called SIT8 for short, so you don't have to remember the full name, just SIT8 is good. So I work on a gene called SIT8, and this gene was actually discovered in a patient with a birth defect called heterotaxy. So let's start with heterotaxy. So what actually is heterotaxy? So if you look at the human body from the outside, our body looks symmetrical. The left side of our body and the right side of our body look the same, right? However, if you look on the inside and you look at our internal organs, the left and right sides of the body are actually not the same. Some organs, like our stomach, if you can see it over here, are located on the left side of the body. Our liver is located on the right side of the body. We have a heart that's kind of located towards the center, but it has its own left and right side, which are different from each other. And so to be able to put all of our organs in the correct places, we actually need to, during development, when the baby is in the womb, the baby needs to actually decide which side of its body is going to be the left side and which side is going to be the right side. So heterotaxy is a disease that occurs when this process doesn't go right. So as I said before, in the normal healthy situation, so almost all of us, we have our stomach and our spleen and our heart kind of towards the left side of our body. We have our liver towards the right side of our body. We have two lungs, one on each side, that are actually different from each other. And some people have a mirror imaged body. So all of these organs are exactly mirror imaged to each other. And these people actually tend to not really have health problems. They tend to be healthy if they're a perfect mirror image. However, we also get patients, we also have children being born where they have some kind of situation in the middle, where it's not the normal situation, it's not a perfect mirror image, but say some organs are on the incorrect side while others are on the correct side, or the organs themselves are not set up correctly along their left-right axes, like their left and right is not set correctly. So this entire range of sort of things in the middle that can go wrong is known as heterotaxy. So heterotaxy is a pretty complex health condition. It affects about 1 in 10,000 people who are born. And the most common symptom of it is actually heart defects. And this is because, as you can see here, yeah, I hope you guys all enjoyed that breakout room activity and you were able to see a little bit better. You're able to see a little bit better what it actually looks like when in real life. I mean, these pictures I showed you earlier, these pictures I showed you earlier, they're very clean and they look very obvious and they're very crisp when we draw a picture like this. But in real life, when a doctor is trying to figure out what's going on with the patient, it can be a little bit more complicated to see what's happening. And I hope you could appreciate that looking at the x-rays. So yeah, so as I said before, I am actually looking at it. So there's a patient that came to our program who has heterotaxy. So their X-ray looked pretty similar to some of the X-rays that you saw in your breakout room. And the doctors and scientists in our program collected DNA samples from this patient. And they were able to then look at the DNA and find what mutations they had. And so they found a lot of mutations in this patient, but one of the mutations they found was in a gene called CYP8. So at that point, the doctors and the scientists passed that data on to me. And I looked at the CYP8 gene and was like, okay, maybe this is the gene that's doing it. So how could I actually know that? So what I went and did after this was I then decided to look in frogs, and I wanted to see if the same gene, if I created a similar kind of mutation that the patient has in a frog, would the frog also have similar kind of health problems? So let me tell you a bit about the frogs that we use. So a lot of developmental biologists like using frogs. They're a very helpful animal for research. So in my lab, we use these two species of frogs. So you can see them here in this left-hand image. So these two smaller frogs at the bottom are one species, and this frog this larger frog on the top is a different species. And these are two species of frogs that we use in my lab. And here I have a little video of some frog eggs. So when a frog first lays its eggs, this is what these eggs look like. And so for a human, our babies develop inside the mother's body, right? They develop in the womb. However, for a frog actually lays its eggs outside of the body and they just develop in water. So we can actually look at the developing baby frog and see what's going on. So here's a nice little video that shows what the first part of that development process looks like. The first part of going from just an egg to a whole animal looks like. So this first part, these eggs, so these eggs are just one cell, and then they start dividing to make many cells. And then those many cells will eventually go to form a full frog, but that takes a long time to happen. All right, let's move on to the next slide. So, so this process that's happening here in this video, this is kind of what the first day of making a frog looks like. After 3 days, so once the eggs are laid, 3 days later, we actually have a tadpole that looks like this. And the reason, one of the reasons that frogs are such a great animal to use for my research is because these tadpoles are transparent. So I can actually see their organs, and I can tell which side of the body their organs are on. So for example, this little tadpole, it already has a heart that's formed, it has a gut, it has a gallbladder, and I can see all of these without having to cut it open. I can just look down a microscope and see this. So I can actually make mutations in frog embryos and frog eggs. So I can make a mutation similar to the patient's mutation in this SID8 gene. So this circle over here is an egg, and I can actually inject this egg with certain chemicals that'll create mutations in this egg in the frog SID8 gene. And then after 3 days, so 3 days after I inject it, we get a tadpole that looks like this. And the tadpole has many organs, but just as you guys in your breakout room activity mostly focused on just the heart, I will also just focus on the heart. I also just focus on the heart in my research, at least most of the time. So in a healthy frog, this heart has, actually, maybe I should go back to this picture of the human heart. So the human heart, this big red blood vessel is called the aorta. So that is a big blood vessel that takes blood away from the heart to the rest of the body. So in a frog, we have a similar thing where the frog actually has a pipe that's also called the aorta that takes its blood away from the heart into the body. And in the frog, this aorta comes out of the frog on the right side of the body and then kind of bends over to the left. And that's what happens in the normal situation. But if the frog has heterotaxy, we can actually get the opposite thing happening, where this aorta comes out on the left and bends over towards the right, or it can just come out in the middle and not bend in either direction. And these two, if a frog has these two, has a heart that looks like this or like this, we say it has heterotaxy. So my main question was, if I give these frogs SID8 mutations like the patient has, do they have heterotaxy? And the answer is yes. So normally frogs that have no mutations in them, most of them do not have heterotaxy. So 1 or 2% of them might have heterotaxy. However, if I make these mutations in frogs, suddenly, so these are 2 different mutations in the SID8 gene that were found in patients. And suddenly, if I make these mutations like this that were found in a human patient, suddenly a lot of these frogs start having heterotaxy, almost 20% of them. So this tells us that this SID8 gene is also important for this birth defect in frogs. And so yeah, so we discovered that that this SID8 mutation causes the same kind of problem in frogs. So now that we know it causes the same kind of problem in frogs, we can use frogs to try and find out what SID8 actually does. So I've told you about the SID8 gene, but I haven't actually told you anything about what it's doing in the human body or in the frog body. And the reason I haven't told you any of that is because I still don't know. So I am still doing my PhD, and the rest of my PhD is going to be me trying to find out what it actually does. But I do have some ideas of what this gene does. So I hope all of you have maybe seen a diagram of what a cell looks like at this point. This is a diagram of what an animal cell looks like. So this animal cell has, for example, a nucleus that contains its DNA. It has a lot of different what are called organelles. So that there are different parts of the cell that do different things. And so there's one part of the cell which you might not have thought about very much, and that's actually the membrane. So that's this membrane on the outside, this border on the outside of the cell. It's like the packaging that everything in the cell is located inside. And so I think maybe that sedate is actually involved at this membrane. And so if you look here in this picture, you can see these little dots, these little circles on that are near this membrane. So what these circles are is that often cells will produce things, produce proteins, produce other kinds of chemicals inside the cell, and it needs to push them to the outside. So it makes things that then need to be let out outside of the cell into its environment. So the way it does that is it'll package these proteins, package these chemicals into these little packages called vesicles. So these vesicles are also made of the same material that the membrane is. Because they're made of the same material that the membrane is, they can actually go and fuse to the membrane and let out their contents on the outside. So I think sedate is involved in this process, but I don't know for sure. That's what my next 3 years of my PhD will be trying to find out. But if it is involved in this process, let me try and ask you guys a question. So what do you think, if a cell produces some kind of chemical and it releases that chemical into its environment, why might it do that? Like, what is the process of, like, why would a cell need to release things into its environment? Things that it made inside itself. Why does it need to push them out? So, food. So trash chute. That's a good idea. So just like, you know, we have things that we need to get rid of from our bodies. So we go to the bathroom. That's how we do it. Cells might do the similar type of thing. That's one idea. But there's other things that a cell might want to get out of its body. All right, so I'll just tell you. So this process is particularly important for cells to actually talk to each other. So a cell can release something into an environment, and other cells that are nearby can pick up that thing and be like, "Ooh, all right, the cell next to me released this. This is some kind of message." So other cells nearby these cells can actually communicate each other by releasing things for other cells to find and pick up. And this is a process that's very important for development, because cells need to be able to talk to each other to coordinate what they're doing in the body to make a body. So yeah, I still haven't found any of this stuff. So I'm working on that for the next couple of years. And maybe if any of you want, you can come to my lab and help me out, because I'll need help doing this. So now I'm getting towards the end of my talk. So now that we've discussed sedate and heterotaxy, I just want to come back to how research like this can start from tragic places, but actually have result in some kind of good in the world. So this research started with a patient who was in a lot of pain, had a lot of suffering due to their health problems. And however, I and my lab mates were able to discover some interesting science. And not only that, we were just able to discover some information that we could tell this patient's parents. And this patient's parents were, it actually was very, very good for them to find out this information. It gave them a lot of comfort. They were happy to know more information about their child. So research like this is very, it has a lot of impacts in a lot of different ways. So now that I've finished telling you about stuff, I just wanted to thank my lab members. So I don't work alone. I work with a lot of people who all help me, and I help them with their research. So we're a big lab, and we do a lot of work like this. And I want to thank the frogs, without whom none of my work would be possible. And I know you guys have been asking questions in the chat, but if anyone else has any questions at the end, please feel free to ask.
Hi, Rita. Yes, everyone has been asking questions in the chat, and we have collected them over the course of your presentation.
Oh, fantastic.
So we'll ask a couple of questions, and if you still have any more questions, feel free to put them in the chat. The first question we got quite early on in the presentation is, what is a birth defect?
Oh, that's a very good question. So there's lots of diseases that someone can be, lots of health problems that someone can be born with, but we don't necessarily call all the different sicknesses or all the different health problems that someone could have a birth defect. A birth defect is specifically some kind of what we call a structural problem. So that means that some part of the body that was supposed to be formed in a particular way didn't get formed that way. So either an organ didn't form in the correct shape or with the correct structure, or your arm or your leg, like a limb, didn't form with the correct structure, or some part of your body basically has some kind of physical problem where things weren't in the right shape, things didn't go in the right location, they have some kind of structural problem. So the structure of that place is a problem, has not formed properly. So other kinds of diseases aren't structural. So for example, some kids might be born with something like a hormonal disease where their body can't produce a particular chemical. So that wouldn't be structural because they still formed all the different parts of their body correctly. It's just some of those parts of the body can't do some of the things they're supposed to do, but they still formed them correctly. Whereas a birth defect is specifically something where a part of the body wasn't formed properly. Is that understandable? Okay.
Yeah, no, I think that was a really great explanation. I myself wasn't really sure like what the distinction was.
Yeah.
And so more about the specific birth defect that we talked about today, we had a question about how exactly are our bodies separated into left and right? Like, how does it know what to do?
Yeah, so that's a very good question. That is a little bit complicated. So there's actually, while our body is developing, so the egg, so hang on, let me go back to the slide where I showed you the frog egg. Okay, so, okay. Okay, so if we look back at this frog egg, so this frog egg at this stage doesn't really have a left or right side. Or, I mean it does, but the left and the right side are the same. Like there's nothing different about the left and the right side. Like if I look at the frog, I can't tell which side is going to be the left side of its body and which side is going to be the right side of its body. So at some point during development, something has to happen for that to change. So the left side of its body has to become different somehow to the right side of its body. And so the way this happens is that about a day after the start of this video, about a day after the embryos look like what they look like at the start of this video, there's actually a part of the body which is called the left-right organizer. And the reason it's called the left-right organizer is it organizes which side of the body is going to become the left side and which side of the body is going to be the right side. So this left-right organizer actually starts to, one side of the left-right organizer will start to express certain genes that the other side does not express. And whichever side, so when I say, okay, yeah, so one side of the body starts to turn on certain genes that the other side of the body does not turn on, or one side of the organizer turns on certain genes that the other side of the organizer does not turn on. And those, because we have different genes that turn on different sides of the body, that can actually tell us which side is left and which side is right. So some genes will turn on just one side, and then that side will go on to become the left side. And other genes will turn on only on the other side, and whichever side that is will go on to become the left. So yeah, a lot of times when people talk about DNA or when people talk about genes, they'll say that these are the instructions for how to build the body. So that's what's going on here, right? Like your DNA has instructions on which side is going to be the left, which side is going to be the right. how do we build the things that need to be on the left side? How do we build the things that need to be on the right side?
Wow, I had no idea that that existed inside of me so long ago. Um,
Yeah.
so it's cool. It's cool to know that that my body had figured this all out like before I was even thinking. But anyways,
Yeah.
now that I can think, I just want to say I thought you had a really great presentation, and everyone else in the chat enjoyed it as well. So thanks again so much for sharing your science with you.